Hello, Dr. Cook and class. I'm Chelsea Seiler, and today I'll be talking about receptor tyrosine kinases and their relationship to cancer. I'd like to start off by explaining what exactly a receptor tyrosine kinase is. They are a group of plasma membrane receptors that show enzymatic activity. They are going to consist of a few different parts. First and foremost, you have the extracellular portion, which you can see over here in this diagram. Then there is the transmembrane helix, which you can see represented in green right here. And finally, there is the cytosolic portion of this particular enzyme, which you can see down here. Now, this portion of the enzyme is the actual portion that has that enzymatic character, because when ligands that you can see up here bind to the extracellular portion, it's on the inside that we're going to see some phosphorylation happening that's going to actually activate that particular enzymatic complex. So the way that this works is once a ligand binds to that enzyme, it's going to cause some dimerization of two local RTKs to happen. And then once that dimerization happens that you're seeing here in this picture, down here in the tyrosine kinase portion of the enzyme, some autophosphorylation between those two segments is going to occur, which will actually activate this portion of the enzyme. And then at that point, other relay proteins in the cell are going to come and bind to this complex down here, triggering a signal transduction to happen, which will eventually cause a cellular response. So here, what we're seeing is several different types of receptor tyrosine kinases next to one another so that we can compare their structure. So they are labeled down here on this portion of the diagram with their different names. But what I'd like to draw your attention to is the extracellular portion of these RTKs, which you're seeing above here. There is a lot of difference in structure in every single one of these RTKs, which makes sense because each of these RTKs has a specific ligand that will bind to it and activate it. If all of the RTK molecules had the exact same extracellular portion, then they would be getting activated by all the same ligand, which is not the case. So it makes sense for there to be a lot of discrepancy among their extracellular structure. However, the portion of this particular enzyme that's within the cell, in the cytosolic portion, most of that is the same. That tyrosine kinase portion is mostly the same, with a couple of exceptions. One that I'd like to note here is this one, the PDGFR. There's an insert in that tyrosine kinase portion, which you can see as a break in the red rectangle representation of that in this diagram. So really, given that the cytosolic portions of these enzymes are all very much the same, that tells us that these are going to be activated similarly. Because all of these enzymes that you're seeing here are going to be activated by that phosphorylation that I was discussing earlier. I'd like to take a second and look at a specific structure of a tyrosine kinase domain of an insulin receptor. So to summarize what we're looking at here, we are looking at the cytosolic portion of an insulin RTK, all right? So all of the red spirals you're seeing are the alpha helices. Now, this particular RTK is comprised of 4 or 5 alpha helices. In this case, we're seeing 1, 2, 3, 4, 5 of them. And those are going to be disulfide bonded to the beta sheet portions that you, or beta strand, excuse me, portions that you're seeing in blue up here. There is one part of this that's kind of hard to see, but the tyrosine containing peptide substrate is actually in pink over here. And again, it's a little bit hard to see, but this is the portion that's going to have those phosphate groups bound to it during that autophosphorylation period after dimerization. So this part is the part that's going to activate that complex and then allow signal transduction to take place. Other things to note about this structure, we can see the nucleotide binding loops over here in yellow, here and here. We can see the activation loop in green there. We can see the N terminus and the C terminus as well represented in this particular image. Next, I'd like to talk a little bit about the function of RTKs. They control quite a bit through different signal transduction pathways within cells. They have a hand in the proliferation of cells, cell differentiation, and of course, cell signaling. You can kind of think of them as a node that communicates information from the exterior of a cell to the interior of a cell. So over here on the left, I'd like to take a look at this diagram. You are seeing here the normal functional activation of a receptor tyrosine kinase. So the left-hand side of this diagram represents the unactivated RTK. You'll see that the ligands pictured in red over here haven't quite bonded to the extracellular portion yet. And another thing to notice about the inactive form is that these two separate RTK molecules here and here are not dimerized. They're not bound together yet. And that's because the ligands have not bound there. But once they do, once those ligands bind to the extracellular portion, that causes that dimerization, that meeting of the two local molecules together. And that, as a result of that, these portions down here, the tyrosine kinase portions, they get brought together and they get phosphorylated. And you can see that phosphorylation happening over here, represented by those greenish yellow ovals with the P's on there. That represents the activated complex. And once you get the activated complex, then other relay proteins that are not pictured here would start binding on, and you'd start to see that signal get pushed to different parts of that pathway. This diagram here that we're seeing on this slide is another representation of how activation of RTKs actually works. I like to include many different pictorial representations of any particular diagram because students will see a ton of different representations, and it's good to really be able to recognize what you're looking at no matter what form it's in. So once again, on the left-hand side here, we're seeing the inactive form, the two separate RTKs not bound to a ligand yet. Once that ligand binds, we see that dimerization happening, those two portions coming together. And then we see that phosphorylation happening, represented by these P's starting to be bonded there. And this happens because ATP is becoming ADP and donating that phosphate into that complex. Now, the difference here in this diagram is that we're starting to see in these multicolored molecules here, we're starting to see those relay proteins start to bind onto that active complex. And then as I go along, you'll be able to see some of the transduction pathways that actually take place. But before I get there, we're going to stop and we're going to do a knowledge check. So assuming that I was presenting this information to students, I would stop and I would have them test their knowledge so far. So my knowledge check that I've embedded into this presentation is to use the word bank below to fill in the appropriate terms to the gray boxes. Some of those terms are used more than once. And then once the students have finished with that, their job would be to summarize what's happening in that diagram in a short paragraph. So a few sentences, just tell me what's happening here. Depending on the level of my students, if it's an AP class in high school or if it was a college class, would depend on how I would phrase this to them. For high school students, I definitely have them work on this in pairs or small groups. For a college class, I might have them do it alone, but it would definitely depend on what I felt their knowledge level was. But since there are no students in front of me right now, I'll just go through this and I will label it myself, as I would do with them and involve them in the process in a classroom situation. So we would go through and we would label together that this here would be the ligand binding site. This up here, this red square represents the ligand itself. The extracellular domain is this right here. This is the transmembrane alpha helix. This is the cytosolic domain. And the catalytic site. And then here, this process with the curvy arrow is going to represent ATP becoming ADP and phosphorylating that activated complex. And then over here, we see the same thing happening on the other side, ATP donating a phosphate and becoming ADP. Once again, how exactly this is going to go class for class depends on many things. It depends on their knowledge level, how comfortable they are, etc. Now I'd like to look directly at a particular signaling pathway that is activated by an RTK. So up here on the top left, we see that yellow kind of two triangle shaped ligand binding to the RTK molecule and having that phosphorylated active complex. But after that happens, after that complex becomes active, those phosphorylated receptor tails are going to recruit a couple of relay proteins, GRB and SOS, which are going to activate the RAS molecule, and then eventually the RAF molecule, which is activated at that plasma membrane. RAF, in turn, is going to activate MAPKK via phosphorylation, which will activate MAPK as well. Now, that phosphorylation happens out here in the normal cytosol, but MAPK can actually go into the nucleus, as we see down here. This is MAPK. And then its job, once it enters the nucleus, is to phosphorylate certain transcription factors, which then will activate gene expression, which we're seeing over here with this DNA molecule right there. But what's important about this and about all of the other signal transduction pathways, because there are a lot, is quite a few of them actually end in expression of certain genes. So if this is all working as it is supposed to, genes get expressed as they're supposed to, but if it doesn't work, sometimes overexpression might mean a gene's getting expressed too much, or if this pathway doesn't work at all, it means the gene isn't getting expressed at all, or not as much as it should be, which can cause a whole lot of problems, one of which is cancer. Our body has a lot of safeguards against cancerous cells. Our body strictly regulates apoptosis, the way cells are growing and proliferating. And if all those processes, again, are functioning normally, then we get normal tissue patterns. However, when they are not functioning normally, then sometimes we get cancer or precancerous cells. Receptor tyrosine kinases largely control those functions. They control cell growth and proliferation and sometimes apoptosis as well. And so if those RTKs are not functioning properly, oncogenic effects may result and then slowly result in cancer. So down here in this diagram, we're seeing a cell cycle diagram with the checkpoints in cell cycle highlighted. So we have the G1 checkpoint here where the cell checks for nutrients, that it's growing properly, and if any of the DNA is damaged. We have the G2 checkpoint checking for cell size and if the DNA was replicated properly, and the metaphase checkpoint, which involves the cell checking for chromosome spindle attachment. Up here in this diagram, we see apoptosis, or the process of apoptosis, which starts up at number 1 with cells that are damaged, stressed, or whatever have you. Once apoptosis is triggered, that cell is going to begin to shrink up, and other proteins are going to get activated to start to break down those cellular components. Enzymes are going to break down the nucleus, and the cell's eventually going to break apart into smaller pieces and eventually will be destroyed or eaten via endocytosis by macrophages. So when mutations occur in genes that code for RTKs, a bunch of things can happen. Some of these mutations could cause gains of function, which would result in the activation of RTKs, even in the absence of their ligand, which is what we're seeing over here in figure B. So what we're observing in this particular diagram is that this complex down here is active, but there is no ligand bonded here. So if a mutation like this occurs, this RTK is active and stimulating that transduction pathway when it should not be, which downstream could result in the overexpression of a particular gene or several other things. Another thing that could happen is amplification of an RTK gene, which would cause there to be more RTKs than there should be. And that increased receptor concentration could potentially be activated more than it should be. So you get the same result there. You get more expression of that gene downstream than you should. Another effect that you might see, or another mutation, is a chromosomal rearrangement, and those can result in an oncoprotein complex of an RTK and its fusion partner. So that's what we're observing over here. We're seeing that these two are fused and dimerized. Again, there's no ligand bonded up top to cause that to happen. And when those two molecules fuse and dimerize, that's what starts that activation process with the phosphorylation and everything there. Also, once that's on, all those relay proteins that start to bond to the active complex will start bonding on and stimulate that transduction pathway as well. You could also see a duplication of a tyrosine kinase domain, which is what we're seeing right here. So in this case, there's not an entire another RTK attached to it. The only thing that is duplicated is that tyrosine kinase domain that you're seeing here. But it does result in the same effect. It results in that gene that, or not the gene, the protein that is activated, that complex is activated and phosphorylated, and then is going to stimulate something to happen downstream as well. Last but not least, we have autocrine activation, which is actually an increased concentration of the ligand, which will then stimulate dimerization and activation of that complex more than it should be as well. So basically, all of these things, all of these mutations can cause RTKs to be more active than they should be, which is going to cause overexpressions of certain genes downstream. So far, 58 RTK genes have been identified, and 30 of those have been linked to specific cancers when they are dysregulated. Specific examples of mutations that can cause that dysregulation would be mutations in the DFG motif and the kinase activation loop, EGFR small frame deletion mutations that might hyperactivate that kinase, extracellular domain mutations specifically are linked to glioblastomas. What happens there is they increase the EGFR protein expression, which then undergoes phosphorylation without ligand binding. So again, we're seeing one of those situations where a ligand is not necessary to stimulate protein or gene expression. Another specific cancer that I'd like to talk about that's linked to RTKs is breast cancer. Overexpression of various RTKs can be linked to breast cancer. Those specific RTKs are EGFRs, epidermal growth factors, vascular endothelial growth factor receptors, so that's the VEGFRs, platelet-derived growth factors, insulin-like growth factor receptors, and fibroblast growth factor receptors. All of these, when they are overexpressed, when they are activated when they should not be, are linked directly with cancer aggressiveness and also the likelihood of disease-free survival. So if you have mutations that affect these particular RTKs, the more serious it is, the more aggressive your cancer becomes, and the less likely you are to survive that disease. So it is a huge issue, and a bunch of research is being done into how to treat that. The next thing I'd like to do is discuss RTK signaling in breast cancer progression. So I'm going to do that using this diagram here, which at first glance seems very, very, very confusing because it's very busy and there's a lot of stuff going on. But the easiest way to look at confusing diagrams like this is to break them apart into things that we can recognize and do know things about. So this line here that you're seeing, this double line, is representative of the cell membrane. And in it, we're seeing embedded all of these red things, which represent RTK molecules. And then we see linked to those RTK molecules, we see these ligands here, 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 and here. Ligands, which I've mentioned before in this presentation. So we have the VEGF ligand, the EGF ligand, the PDGF ligand, and the FGF, okay? So when those are bonded, when those RTKs are active, we see different signal transduction pathways happening. So this over here, the VEGF ligand is going to bond here and activate this particular RTK, which will then start the JAK-STAT pathway, which then after that induces the SOX2 expression, which we see down here. And once that is activated and expressed, or I should say overexpressed, that can lead to CSCs or cancer stem cells. Here in the middle, we're seeing the EGF ligand activating this particular RTK, which if you look is actually linked to a few different things, the MAPK pathway, STAT as well. But again, the end result is cancer stem cells once again. The PDGF pathway is linked directly to STAT here again, which is going to then be linked to gene expression and leads to cancer cells. Over here, we have cyclin D1 being linked to the FGF ligand, that expression, the MAPK pathway again. So really what I'm getting at is all of the overexpression of these particular RTKs leads to cancer. Now, given all of that information, given that there are so many pathways and so many different RTKs, what I would have my students do after looking at a diagram like that is do their own research. I would have them pick a type of receptor tyrosine kinase, any particular type. Now, if they're younger students, if they were an AP class in high school, I would give them a list to pick from. If they were older students, college students, I would have them look up their own. But ultimately, what I would want them to do would be research how that particular RTK might be linked to a specific type of cancer. And in about 500 words, again, I could extend that if it was an older group of students, I would want them to talk about the normal functional mechanism of that particular RTK, what signal transduction pathways is it linked to, what is its normal functionality. Then I would want them to talk about the mutations that can lead to dysfunctionality of that RTK, what specific genes are mutated, how does that affect the RTK itself, does it change how the RTK looks, functions, what? And then finally, I would want them to discuss the mechanism by which that RTK's normal function is interrupted. So if it is a structural change, then how is that structural change affecting the functionality of that RTK? Does it make it overexpressed or underexpressed, or what is happening? And then again, depending on the group of students, we might present our findings to the class. So this could be turned into their own presentation project, or it could be just a small paper or something like that. But I would like to thank all of you for listening to my presentation. Have a great day.